If I were to ask you what antihistamines do, you'd probably say something about allergies, right? As someone who keeps a bottle of Claritin in all my backpacks, that's what I thought too. But it turns out that antihistamines are everywhere. They're in popular ulcer medicines, they gave rise to antipsychotics, and totally a surprise to me, they played a role in the development of motion sickness medicine. So, today's story comes in 3 parts. How did scientists discover that histamine had such an important role in allergies? How did pharmaceutical researchers find good drug candidates to block histamine? And finally, how did antihistamines find their way into so many drugs? Naturally, before we talk about antihistamine, we need the story of histamine itself. By the first decade of the 20th century, doctors had documented the symptoms of both allergies and full-blown anaphylaxis, but they didn't know exactly how they worked. Totally separately, chemists had figured out how to synthesize histamine in the lab by cleaving off the carboxyl group off of the amino acid histidine. That's where the molecule gets its name. It's an amine derived from histidine, hence histamine. But at the start of the 20th century, nobody knew what histamine actually did in the body. That work was started by this dapper gentleman, Sir Henry Dale. In 1904, he took a research job at Burroughs Wellcome where he and a colleague started studying a fungus called ergot. Scientists had seen ergot stimulate contractions of the uterus before, so the two of them hoped that they might create a drug from ergot that could speed up labor. But the research had a different impact. It turned out that the stuff in the fungus behaved really similarly to histamine, what they called beta-I at the time. Among other things, both ergot and histamine led to uterine contractions in animal experiments. And since he'd figured out how to isolate histamine from ergot, the next thing was to figure out what exactly it did in the body. And that meant injecting it into different animals and measuring what happened. For example, he shot some into a frog and it went all slack-jawed, quote, succeeded by depression of the central nervous system for periods increasing with the dose. Then when he gave it to rabbits intravenously, their breathing and heart rate would become irregular, then eventually return to normal again. But if he gave them a bigger dose, their symptoms would get even worse, and they'd experience severe obstructed breathing before dying from heart failure. He noticed the same labored breathing in guinea pigs. At the very end of the paper, Dale mentioned something that he'd become obsessed with for the next few years. When they gave animals histamine, some of their symptoms were kind of like what happened in anaphylactic shock, that severe form of allergies. But they still weren't sure how it all fit together. Dale had extracted histamine from mucous membranes and certain tissue samples, but there was a chance the tissue was just contaminated with histamine. They weren't sure the body created it. Fast forward to 1927, and a research group comprised of Dale and a few others, including the co-discoverer of insulin, Charles Best, observed that vertebrates do produce histamine on their own, especially in the lungs. At which point Dale was like, oh yeah, knew it all along. And this meant that histamine had some kind of function in our normal biology, but its role in allergies was still unclear. Just a few years later, in 1931, a Japanese scientist named Kei Watanabe did an experiment where he'd measure histamine in guinea pig lungs, induce allergies, then measure histamine again. He measured more histamine after allergy than before, which was evidence that histamine was somehow involved in the disease process. Here's our modern idea of how it works. Allergies are kind of like a misfire from the immune system. The first time you run into an allergen, like pollen or peanuts, your immune system classifies it as an invader and creates immune cells that remember it and lead the attack the next time they see one. Then, when that allergen gets into the body again, it binds to antibodies on special immune cells like basophils and mast cells, and once those cells are stimulated, they release a bunch of different inflammatory molecules all at once. And one of those molecules is histamine. So let's say that an allergen lands in the nasal cavity. Immune cells will rush to the scene and start the inflammation process there. That causes the release of histamine, which results in leaky blood vessels and a runny nose and congestion. Now, if the immune overreaction is even more severe, it can create a potentially lethal allergic reaction called anaphylaxis. Check out my video on the history of EpiPens for more on that. Either way, whether it leads to full-blown anaphylaxis or more mild allergies, histamine is such a big deal because it's involved in that inflammatory overreaction. But histamine itself has a few other roles around the body. Like, it can be produced by multiple types of immune cells, and it can also be a neurotransmitter, one of those chemical messengers between nerve cells. But coming back to the story, now it's the 1930s, and doctors knew there was this chemical called histamine involved in allergic reactions. But all of this histamine research was happening during a shift in our understanding of pharmacology. See, up to this point, some scientists thought that drugs worked more through brute force. They thought drugs could physically rip apart cells or implode them from the outside in. But around the late 19th, early 20th century, scientists started hypothesizing that cells had specific receptors on them, and when specific chemicals fit into those receptors, they triggered some kind of biological action. It was kind of like lock and key. And this model would become, well, the key to finding allergy medications. Because if you knew what the chemical key looked like, and you knew how it fit into its receptor lock, then you could find a different key that fit into that same lock. We'll focus mostly on the H1 histamine receptors in this video, and these guys are found all over the body, especially in smooth muscle, in the lining of our nose and airways, and in the central nervous system. So in this classic receptor model, there are two big factors that they cared about: affinity and intrinsic efficacy. Affinity is how well a drug binds to its receptor, while intrinsic efficacy is the ability of that drug to trigger a physiological response from the receptor. To come back to our analogy, affinity is how well a key gets to its lock, and intrinsic efficacy is how well the key unlocks the lock. And back in the early days of this model, scientists thought there were two things a drug could be given these two factors. A drug could be either an agonist or an antagonist. Agonists stimulate a receptor and trigger some kind of effect. For example, opiates like morphine bind to their receptors and elicit the same effect as if your naturally occurring endorphins were to plug into those receptors. Using those terms from earlier, we'd say that agonists have high affinity for their receptor and can have varying degrees of intrinsic efficacy. And in general, drug developers prefer higher affinity, but they don't always want high intrinsic efficacy. That's where antagonists come in. They bind to a receptor, they have affinity for it, but they have no intrinsic efficacy, and that prevents other chemicals from triggering that receptor. It's kind of like jamming a key into a lock so that you can prevent other keys from getting in and unlocking it. There are a couple other ways that molecules can affect receptors, and I'll talk about some of those later. Either way, this basic receptor model gave early 20th century scientists an idea of how different drugs might work. And by the 1930s, scientists had enough information to find an antagonist to histamine. This is Daniel Beauvais, a researcher at the Pasteur Institute in Paris. One of his predecessors at the PI, Ernest Fourneau, had put together a catalog of potential anti-malarial compounds. So Beauvais and his colleague, Anne-Marie Staub, who unfortunately I couldn't find a photo for, started screening the compounds in that catalog for antihistamine properties. And one of those drugs, piperoxine, did block the effects of histamine in guinea pigs. And another one, codenamed 929F, blocked anaphylaxis. Unfortunately, they were both too toxic for human use and didn't lead to a marketable drug. But it was a good starting point. Beauvais and Staub were able to show that you could tamp down allergy symptoms by blocking histamine, so antihistamine drugs were a possibility. Now, elsewhere in Paris, there was a scientist named Bernard Halpern working for the pharmaceutical company Rohan-Poulenc, and he described an antihistamine drug called fenbenzamine. And not only was it more potent than Beauvais and Staub's drug, it was safe for humans. So in 1942, Rohan-Poulenc released it under the brand name Antergan. Now, it was World War II, and Halpern was Jewish, so unfortunately, he had to flee for Switzerland for safety at this point. But Beauvais and the team at the PI were able to collaborate with his team and keep the research going. And in 1944, they published their research on a safe and effective antihistamine compound, pyrilamine, or what's also called mepyramine. Rohan-Poulenc sold it under the name Neo-Antergan. And it turns out that they were onto something with that receptor model from earlier. Antihistamine drugs like pyrilamine do fit into the same lock as histamine. Now, those scientists in the 40s imagined that antihistamine drugs acted as neutral antagonists. High affinity, no intrinsic efficacy. Basically, they sit in the receptor like a big dumb rock and block histamine from causing allergy symptoms. But these days, we know that H1 receptors can actually exert their effect even without a compound in them. They're what's called a constitutive receptor. It's kind of like a leaky faucet where there's always a little bit of water coming out. In this analogy, an agonist is a wrench that can turn the handle and cause more water to come out of the faucet. Meanwhile, a neutral antagonist would be like a piece of clay that gloms onto the handle and keeps wrenches from turning it. But it turns out that there's another option. A wrench can turn the handle the other way and stop the flow of water past its baseline. This is called an inverse agonist. It has an effect. It is an agonist, but in the opposite direction as a traditional agonist. Coming back to the story, it's 1942, and World War II is making it hard for American scientists to read all this French antihistamine research. So separately from all of them, a professor at the University of Cincinnati was conducting some research that would lead to one of the best-selling antihistamines of all time. The professor's name was George Revachol, and he was interested in chemicals he could use as antispasmodics, or muscle relaxers. And one of the best they had at the time was scopolamine, but it came with some serious side effects on the nervous system since it affects acetylcholine, an important neurotransmitter. Revachol's lab got to work finding an alternative, but over the course of their experiments at the university, they realized that one of their drug candidates happened to be really good at blocking the effects of histamine. Soon afterwards, Rivichel moved on from academia and got a job at a Detroit-based pharmaceutical company called Parke-Davis. And this is where he decided he'd test his new compound, what they called diphenhydramine. After he and a handful of other researchers tested it on guinea pigs, not only did it calm down smooth muscles, but it was indeed an effective antihistamine with a good safety profile, which meant it was time to get this thing into pharmacies. But since Rivichel created the drug before working for them, Parke-Davis would have to buy it from him before they could sell it. So they offered him 5% royalties on the sale of the drug over the life of the patent, which of course is a pretty sweet deal, so Rivichel agreed and submitted a patent in April of 1944. Parke-Davis started marketing this benzhydryl ether in 1946 as Benadryl, and they advertised this thing heavily, more than any other antihistamine at the time. And Benadryl's sales success kicked off an explosion of antihistamine development, which gave us drugs like promethazine, which you might know as Phenergan, or doxylamine, which is the antihistamine in NyQuil, as well as chlorpheniramine, which we'll come back to in the next section. But by the mid-1940s, it was clear that this first generation of antihistamines came with some side effects, like fatigue, dry mouth, nausea, dizziness, and the big one, drowsiness, or sedation. Some people even reported passing out and feeling disoriented after taking it. It turned out that these first-generation antihistamines cause drowsiness because they can cross the blood-brain barrier, the membrane that regulates what kind of things cross from the blood into the tissues of the brain. From there, they plug into H1 receptors in the central nervous system and interfere with the arousal part of circadian rhythm. Since drowsiness was such a problem, a drug company called G.D. Searle said, okay, what if we were to combine diphenhydramine with a stimulant to counteract the drowsiness? Unfortunately, this new compound, dimenhydrinate, didn't quite succeed at the non-drowsy part. But there was an unexpected effect. In 1947, a doctor from Johns Hopkins prescribed dimenhydrinate to a pregnant woman for her allergies, and while it didn't help, the woman noticed that when she took it before getting on a streetcar, she didn't get car sick like she usually did. So G.D. Searle decided to test whether the drug was actually effective at preventing motion sickness. They organized a trial where they gave dimenhydrinate to troops crossing the Atlantic Ocean and found that it prevented nausea better than a placebo, and as expected, the only side effect was drowsiness. They marketed this anti-nausea drug as Dramamine, which of course is still around today. But still, it must have been so frustrating in 1950 to have a medicine that relieved allergy symptoms, but made it impossible to get through the workday without taking a nap. Luckily, throughout the 60s, scientists started the search for a second generation of non-drowsy antihistamines. The first successful non-drowsy antihistamine was terfenadine, marketed by Hoechst, Mary & Russell under the brand name Seldane. It came out in Europe in 1981, then in the US in 1985. And soon after it hit the American market, it became the #1 prescription drug of the year, and eventually reached $800 million in revenue. Not quite blockbuster status, but still a good example of what you can do with a monopoly on a drug category and high demand. One of the companies that tried competing with Hoechst was an American pharma company called Schering-Plough. They were already selling a first-generation antihistamine called chlorpheniramine when Seldane came to the US and took over the market. So scientists at Schering-Plough responded by developing loratadine at the end of the 1970s and filing a patent in June of 1980. But they were still over a decade away from actually selling anything. See, pharma companies usually patent their drug before clinical testing so they can research it without worrying about someone reading their research and beating them to market. If they're going to spend over $1 billion developing something, they want to protect their investment. The patents themselves run for 20 years, but since the patent is taken out before clinical testing, the manufacturer doesn't have a drug to sell right away. So the more time they spend on trials and filing for FDA approval, the less time they have to sell the drug within the patent window and charge higher prices. Plus, clinical trials cost hundreds of millions to billions of dollars, so time is money. Loratadine's preclinical trials in animals went well, as did phase 1 human trials for safety. But then during phase 2 trials, which involve a bigger group of humans to determine effectiveness, they switched from capsule to tablet form. And that process took about 2 years before they were able to progress to phase 3, which is the most expensive part of drug trials. Schering-Plough recruited thousands of patients for a double-blinded randomized controlled trial, and participants reported improved allergy symptoms with loratadine compared to a placebo. Schering-Plough filed for FDA approval on Halloween of 1986 and waited. Of course, not everyone was convinced right away that it was such a good drug. The year after they filed, a medical reviewer at the FDA wrote that Schering-Plough's proposed dose of loratadine didn't seem effective compared to a placebo. He said that loratadine itself was an effective antihistamine, but the 10 milligram dose didn't lead to significantly better results than a placebo. In his medical officer's review from November of 1987, he proposed that 40 milligrams would be the lowest effective dose. The problem, though, was that higher doses were more likely to cause drowsiness, and that wouldn't do Schering-Plough any good. They wanted to market a non-drowsy antihistamine, and they thought they had it, a balance between safety, efficacy, and non-sedating effects at 10 milligrams. And in May of 1988, the greater FDA agreed with them. The 10 milligram dosage was no more sedating than a placebo, and in September of '89, an initial FDA report made it seem like formal approval was coming. But this is when the story becomes a roller coaster. Because that same year, the FDA reorganized its personnel and someone new took over the loratadine case, which delayed approval even further. And since there was already an approved non-drowsy antihistamine in the form of Seldane, loratadine was low priority. But then throughout the early 90s, stories started popping up from people who had taken both Seldane and certain antibiotics or antifungals and developed potentially dangerous heart arrhythmias. This was enough for the FDA to reevaluate Seldane's safety. And in 1992, the FDA told their manufacturer to put a warning label on the drug. And this was great news for Schering-Plough, because their 10 milligram loratadine was the obvious alternative. The FDA granted loratadine approval in April of 1993, which Schering-Plough marketed under the brand name Claritin. But at this point, they only had 7 years of patent protection left, so they just go nuts marketing it. And this happened to line up with new rules around drug advertising. Pharma companies were already allowed to advertise directly to the consumer, but in 1997, the FDA relaxed its rules on TV advertising a bit. This is when the whole "ask your doctor if Claritin is right for you" became a thing. So Schering-Plough spent over $300 million to advertise Claritin on television, including sponsoring the 1999 World Series. By the way, Claritin wasn't the only one going hard on advertising. Drugs like Viagra and Prilosec had big direct-to-consumer TV campaigns at the time too. But according to a Wall Street Journal article, Schering-Plough spent more marketing dollars on any single medicine than any other company in history up to that point. And oh boy, did it pay off. Claritin quickly became the most popular antihistamine and the best-selling. It hit blockbuster status in 1997, only 4 years after approval. According to a 2001 article in the New York Times, sales for Claritin products earned Schering-Plough $1.4 billion in 1997 and $2.6 billion in 2000 after the campaign. And other non-drowsy antihistamines followed. Like Pfizer came out with cetirizine, also known as Zyrtec in the US. And after Seldane's sales went downhill, Hoechst, Mary, and Russell started selling a metabolite of Seldane called fexofenadine, otherwise known as Allegra. Shortly afterwards, they merged with Rhone-Poulenc from earlier in the story to become Aventis, now Sanofi Aventis. So what was the secret? What made these drugs non-drowsy? Well, these new antihistamines still occupy H1 receptors, but they don't cross the blood-brain barrier as easily as the first-generation drugs do. Therefore, they won't affect those neurons in the central nervous system that trigger alertness. The other big difference is how our bodies clear them from the brain. One of the mechanisms for doing that is a transporter called P-glycoprotein, and this thing can be a big deal when it comes to how much of a drug is absorbed or not. First-generation antihistamines don't really interact with P-glycoprotein, so they don't get kicked out as easily as the second-generation drugs do. Alright, now all this time I've been specifying the H1 histamine receptor, which means that at some point scientists discovered more than one. So how did that happen? Well, antihistamines had all these unexpected effects for the entire time scientists had been studying them, and in the 1960s, a handful of them started focusing on why that might happen physiologically. Like, one experiment published in 1960 showed that histamine had an effect on cardiac muscle, which meant that the tissue had histamine receptors, but when they tried to block that receptor with an existing antihistamine drug, it didn't work. If this were a histamine receptor, the antihistamine key should block the lock, but it didn't. So they hypothesized that maybe this was a new type of histamine receptor they hadn't identified yet. And a few years later, other scientists noted the same thing but with stomach acid. They could stimulate gastric acid production with histamine, but couldn't inhibit it with the traditional H1 antihistamine. They concluded that, quote, "The relative activities of several histamine analogs support the differentiation of histamine receptors into at least 2 classes," which meant that any histamine receptor that could be blocked by existing antihistamines should be classified as H1 receptors, but histamine receptors not blocked by those drugs should be known as H2s. Now remember, this experiment started with stomach acid, so now that they knew more about these new histamine receptors, scientists could more easily find an antagonist to it, which would inhibit its action and inhibit stomach acid secretion. And there are 2 common conditions that would benefit from that inhibition: stomach ulcers and GERD, or gastroesophageal reflux disease. Enter Sir James Black. In 1964, he joined the company that would eventually become GlaxoSmithKline, and started looking for an antihistamine that would work on these hypothesized but yet to be documented H2 receptors. His team settled on a compound called burimamide. It blocked histamine from stimulating its receptors in the stomach, plus a few other places around the body, and while it never became a marketed drug, by antagonizing this specific receptor, it showed that H2 receptors actually existed. It was the key for a lock they'd only hypothesized up to that point. After another decade of clinical trials, though, their company landed on cimetidine in the early 1970s, and this thing turned out to be really good at reducing gastric acid, which made it a great treatment for stomach ulcers. Since it was a histamine antagonist called cimetidine, they released it under the brand name Tagamet, and right away it was a hit. In 1986, it became the first drug in history to earn $1 billion in annual revenue, which means that the very first blockbuster was an antihistamine. The success with Tagamet led to more H2 receptor antagonists like famotidine, which you might know as Pepcid, and ranitidine, which you might know as Zantac. And while H2 antagonists are still around, proton pump inhibitors are more often the go-to drug for GERD and stomach ulcers these days. But H2 wouldn't be the last histamine receptor. By the mid-1970s, researchers had years of research on the neurological effects of antihistamines, so there were almost certainly unfound histamine receptors in the brain. Sure enough, research published by French scientists in 1983 showed evidence for new H3 histamine receptors, and by the early 2000s, scientists were studying H3 antagonists for neurological disorders. Then, once the Human Genome Project data came out, multiple research groups found a fourth histamine receptor all around the same time. H4 receptors turned out to be the most concentrated in the immune system, and have a role in mediating inflammation, which might help scientists treat asthma better, among its other immune functions. But there was another effect of antihistamines that I haven't really talked about yet. See, scientists wondered if the sedating effects of first-generation antihistamines might calm people down before surgery, and when they were looking into potential candidates for this indication, their antihistamine research resulted in the very first antipsychotic medication, chlorpromazine, what we call Thorazine in the US. That story didn't quite fit into this video, so I made a separate video all about it available exclusively on Patreon. If you click here and sign up, you'll get access to all my exclusive content and support this channel in the process. And if you're not in the spot to support financially, consider sharing this video with a fellow allergy sufferer or checking out some of my other videos. Either way, thank you for watching. I'll see you next time.